Hey, welcome to Bio 100. Today we're going to jump in and kind of talk a little bit more today about pedigrees and who's your daddy, believe it or not, we know, and DNA fingerprinting, kind of talking about what you've seen and maybe some of these CSI type shows, Criminal Minds and beyond. We'll kind of talk about how each one of these fits and what we can do. But first, I think pedigrees will be a little more interesting to some of you guys. So what is a pedigree? Well, us as being LDS, for the vast majority of us being LDS at least, we understand that pedigrees are kind of this genealogy with a spin. So it's essentially a family tree that shows the inheritance of traits. Why is this important? Well, genetic counselors can use a pedigree to determine if couples will produce healthy children. And actually, you could do this too. It could be a fantastic thing to do on a first date or second or third date, to sit down and figure out what kind of genetics your children are going to have due to inheritance patterns in your own families. But disease can be traced across a pedigree, and that's what's really interesting about this. So we've talked about sickle cell anemia a little bit, hemophilia. These are all things that can be traced on a pedigree. Other traits can be examined as well. So believe it or not, right, like things like having a cleft chin, hairline, eyebrow, size and shape, eyelash length, these are all things that can be looked at on a pedigree. One interesting one is even tongue rolling. The way that you fold your tongue can actually be dominant or recessive. And I wish we were in class for this, because there's always a handful of students who cannot roll their tongue at all, and some students who can do like some really funky shapes. Who knew that was genetics? So how do you read a pedigree? So since you're looking at traits through several generations, you want to have a way to do that. And there's some really common symbols. Okay, so looking here at the right at the first circle, so circles are going to be female, squares are going to be male. And then if they're married, there's going to be a line that connects them, married or mating, because you can also do pedigrees in things outside of humans. And then offspring in birth order, from left to right here. And then affected individuals are going to be fully crossed out or colored in. And then individuals that are carriers or thought to be carriers are going to be half colored in or hashed in. Okay, so here's your first shot. What I need you to do is maybe take a picture of this slide with your phone, or quickly hit the pause button and write it down. And then what you want to do is take this as an example and be able to, actually, you don't need to take a picture, write it down. Some of that information on the pedigree analysis is below here on the bottom right. But on the left here is a list of people, and you should be able to create a pedigree from this. So I'm going to go ahead, ask you to hit the pause button and take about 5 minutes to actually draw this out. Now, there's no carriers, there's no infected people here. You just need to draw the pedigree out. Okay, if you haven't done it or haven't finished it, please do take the time to do it. It's pretty important. And let's kind of go ahead and move on. So this is what you should have drawn out. And don't worry that the boxes are filled in. That's just showing gender essentially here. But you've got Fred and Norma that are married, Steve and Karen and Bob and Ann that are all married, and then their children listed left to right from oldest to youngest. Okay, actually, and I should also mention that Karen and Ann are also written out. Karen and Bob are also from left to right in age as well. Okay, so pedigree can help you figure out if a disease is recessive, dominant, or X-linked. And what I want you to do is I want you to look at this figure right here and tell me whether this disease that we're looking at, or this abnormality, in this example polydactyly, which means multiple fingers or toes on hands and or feet. I want you to figure out if this is dominant, recessive, or X-linked. Go ahead and work that out. Okay, so as we work through this, polydactyly, it's where you have an extra finger. It can also be extra toes. And then when we look at the pedigree, what did you learn about it? Is it sex-linked? No, it's autosomal. Is it dominant? What do you think? Yes, it's a dominant trait. So polydactyly is a dominant trait. Okay, hopefully you were able to figure that out. And there's a lot of hints in there, right, that if you have two parents that are polydactyly, they can have non-polydactyly offspring. That should let you know that it's that the homozygous condition, at least, right, doesn't have to have it. All right, so attached earlobes. You might want to reach up right now. Are your earlobes attached or not attached? You can figure that out. Okay, I want you to just take a few minutes and look over this pedigree and tell me, is this a dominant, recessive, or sex-linked trait? Okay, hopefully you actually took the time to do it. Let's keep rolling here. So this is an autosomal recessive trait. Did you follow that and figure that out? You have all the affected individuals when they have attached earlobes are recessive, right? You see that all throughout the pedigree, so that makes it pretty straightforward. Here's another one. I want you to look at this and tell me, is this dominant, recessive, or sex-linked? Okay, so this is a sex-linked trait. This is muscular dystrophy. The pedigree is is an X-linked trait. Who are the carriers here? Now, we didn't list that, and maybe that threw you off a little bit, but look at it right now. Who's the carrier? It should be pretty obvious, especially because you know it's X-linked. Yeah, so it turns out that mom in this situation is the carrier. Okay. So this is one where I'm actually just going to turn you loose for a while. What I want you to do is there's two different pedigrees, right? So you have pedigree A that's very different from pedigree B, and I want you to answer questions A through D for both pedigrees. And the answer is just going to be yes or no. So you look at pedigree A, could this trait be inherited as a simple autosomal recessive? Okay, so you look at that. Is that a yes or a no? And then look at pedigree B, is that a yes or a no? And then go down through the rest of these. And I want you to hit the pause button and really work on this and figure out if you can answer those questions A through D for both of these pedigrees. Okay, so hopefully you're back and you've actually worked through these. I'm gonna kind of put up some information here. You can kind of see how the genetics almost certainly worked out here. And you should, this is kind of like that repeat that we did a couple of lectures ago where we looked at rabbit genetics and a few other genetics, right? That you should be able to figure out who has what. So for example, if the offspring have both phenotypes in them, right, that you know that the parents have to be heterozygous. So in both of these, the parents are going to be heterozygous. Okay? But sometimes it might not be clear whether an individual is heterozygous or homozygous dominant, right? Because the dominant will play out either way. Okay, so here's some other examples here. If they were X-linked or sex-linked, this might be how those would play out. And then here are the answers that you would get. Now, I'm going to invite you to, I'm not going to cover these because I want you to struggle with this. I want you to figure out why one's yes and why one's no. The answer, everything you need is right here. It should be pretty obvious. If it's not obvious, I would invite you to meet with a TA and go through this together. Okay, but what I'm going to do right now is just keep on going. And what I'd like you to do is hit the pause button again and work through this on your own and see if you can figure out how purple ears work in a pedigree of a family. Okay, so what do you think? Is this autosomal dominant, autosomal recessive? Is it sex-linked? What could be happening here? Okay, if you haven't drawn it out, hit the pause button before I advance and do. All right, I'm about to advance. Here we go. So this is kind of what it looks like. So it actually could be autosomal recessive or dominant. You don't exactly know. And so I hope you're able to kind of sort that out. And some of you guys might be thinking, well, this is super tricky. Are you going to pull this kind of shenanigan on the exam as well? And the answer is no. We're not going to do that to you. If there's both possibilities there, we're going to expect you to be able to defend your answer and tell us why you got what you got, and then we'll give you the points that add up to your response. We're not tricky here. We're just trying to show you that it's not always super straightforward as to what's recessive and dominant. Okay, so DNA fingerprinting, what is it? So it's used in forensic science. It's really kind of like a human barcode. Each one of us has our own barcode inherent in our DNA, kind of like a fingerprint. And it helps in the identification of individuals. So like things like paternity testing, criminal cases, you can use DNA to go back and figure out who's the father, who committed what crime. And no two individuals are genetically identical except for monozygotic twins. Okay, those are identical twins. And differences can be found within the DNA of every single individual who ever lived, except for monozygotic twins. Okay, so here's how this kind of works. So in the first step, you're going to isolate DNA from a tissue sample. And the DNA can really come from any sort of tissue, like hair, bones, skin, blood, anything like that. DNA is cut into fragments with an enzyme, and then those excuse me, the DNA from the different individuals really produce fragments of different sizes. Remember, so the DNA isn't all the same. If you have an enzyme going in and cutting DNA at a set place, that just because we're all different, our DNA lengths will be different. Hopefully this will make sense in a minute. The fragments are going to separate based on size and when you visualize them. And each person's set of fragments is going to be unique, right? Our DNA is unique, so if we're cutting that DNA in very specific ways, then we all should have a different fragment set. One of the things we do as well is we amplify the DNA. So this is PCR chain reaction. You can follow this link and watch it. It's very in depth. It's very good. And essentially what we're doing in PCR is we're using primers, polymerase, and these machines that heat and cool the DNA to allow us to make lots and lots of copies of a region of DNA that we're interested in. And I would highly recommend that you follow this link. I'll put the slide up on Learning Suite, and you can follow that link. But any link that will teach you about PCR or polymerase chain reaction, which is just amplification of the DNA, will do. So step one, isolating DNA and amplification. So a small amount of DNA can be amplified. This can be really, really small. This is how forensic science works, right? That you can find the tiniest blood spot, and you can get DNA out of that, and then you can amplify it using polymerase chain reaction, which is these set of steps here. But I highly encourage you to watch a YouTube video on it, following this link or another on PCR. Okay. So then the DNA is mixed with specific ingredients needed to make copies. So in a PCR tube, right? So we're doing polymerase chain reaction here. You're going to mix it with things like nucleotides, primers. These primers are very specific regions of the DNA, Taq polymerase, and then it all gets heated. The heating splits the DNA molecules into two complementary strands. The Taq polymerase builds a new complementary strand using the primers as an indicator of where the Taq polymerase should start generating DNA and the nucleotides. And then the DNA is heated again, it's split, and they do a series of cycles. And after about 30 cycles of DNA amplification, you go from essentially a single gene up to over 1 billion copies. And then you take those 1 billion copies of DNA and you're going to cut them. And you cut them with what we call restriction enzymes. Now, restriction enzymes cut DNA at very specific places and in specific ways. So if we look here on the far left-hand side at the top, you have EcoR1. Okay, this is a restriction enzyme that was actually purified from E. coli, which is the source organism, and that restriction enzyme cuts DNA everywhere where you have a GAATTC. It will cut between the G and the AATTC, and you'll get these overhanging fragments of DNA. So then if we go down, you can see PST1. This comes from this other type of organism here. It also cuts an overhang type cut, but it cuts in a different spot. So EcoR1 cut at GAATTC, PST1 cuts at CTGCAG. And then the last one there, if we're looking at SMA1, different organism that does what's called a blunt end cut. So there's no overhanging DNA here. It's cutting right in between CCC and GGG. And anywhere in the DNA where that restriction enzyme comes in contact with that sequence, CCC, GGG, it will cut. Okay, so the DNA is going to be cut using these fragments from restriction enzymes, and as it cuts through these sites, there's going to be a lot of different variation, and so it'll cut each fragment into 2, 3, 4, 5, 6, 7 different types, different sizes, and that's all dependent on how many times that restriction enzyme comes in contact with the sequence that it cuts. So then the last step is visualization. So you're doing gel electrophoresis, which really separates the DNA based on its size. So you run an electric current through an agarose gel. Now you can think about an agarose gel, it's really just like super high-end Jell-O. That's basically what it is. We're LDS, right? The vast majority of us, so we should know what Jell-O is. And essentially what happens is the small fragments, so those fragments that were very small, that were cut from our restriction enzyme, will run through the agarose gel much faster than those fragments that are bigger. And that makes sense if you think about it, right? Things that are small are going to squeak through faster than things that are larger. Okay, so then these fragments, once they've been pushed through these agarose gel, they can be transferred to a filter paper, and then you can label the DNA with a probe, and that will tell you where each fragment is in the gel. And then you get things that look like this on the right, where you have the male we have this figure on the right here, and then you have the male on the left, and then there's child 1, child 2, and then the female. And each person will have a unique pattern of bands. We've been over that a few times now. And each band must be present in the child if it came from the parents. Okay, if it came from. Okay, and so here's I hope that was clear, right? Each band is present in a child must also be present in one of the child's parents, right? Otherwise, that child is not from one of those parents. And let's kind of illustrate that here. So I want you to look at that with this rule, right? What conclusion can you draw from this test? Is Mary and Bob, so you have Mary, the mother of the child, and then you have Bob and Larry. Is Bob or Larry the father? And is Mary for sure the mother? hit the pause button and work through that. Okay, let's come on back. So what do you think? Who's the father here? Okay, so if you look at that, it has to be Larry, right? So Mary shares the bar with child, and it shares both of her DNA fragments with the child. So Mary's definitely the mother. But if you look at Bob's first DNA fragment is lacking and does not overlap with the child, whereas all three DNA fragments of Larry's does overlap with the child. So meaning that Mary and Larry share parentage over this child. And that's all we're going to cover today. So hopefully that made sense. I hope that you actually did take the time to pause as you went through there and work through those. It's really important. I hope you're staying safe and having a good time out there, and that everything's going well for you. Have a good one.